Chapter 8 Control of Gene Expression. An organism's DNA encodes all of the RNA and protein molecules that are needed to make its cells. Yet a complete description of the DNA sequence of an organism, be it the few million nucleotides of a bacterium or the few billion nucleotides in each human cell, does not enable us to reconstruct the organism any more than a list of all the English words in a dictionary enables us to reconstruct a play by Shakespeare. We need to know how the elements in a DNA sequence or the words on a list work together to make the masterpiece. In cell biology, the question comes down to gene expression. Even the simplest single-celled bacterium can use its genes selectively. For example, switching genes on and off to make the enzymes needed to digest whatever food sources are available. And in multicellular plants and animals, gene expression is under even more elaborate control. Over the course of embryonic development, a fertilized egg cell gives rise to many cell types that differ dramatically in both structure and function. The differences between a mammalian neuron and a lymphocyte, for example, are so extreme that it is difficult to imagine that the two cells contain the same DNA. Figure 8.1. For this reason, and because cells in an adult organism rarely lose their distinctive characteristics, biologists originally suspected that genes might be selectively lost when a cell becomes specialized. We now know, however, that nearly all the cells of a multicellular organism contain the same genome. Cell differentiation is instead achieved by changes in gene expression. Hundreds of different cell types carry out a range of specialized functions that depend upon genes that are only switched on in that cell type. For example, the beta cells of the pancreas make the protein hormone insulin, while the alpha cells of the pancreas make the hormone glucagon. The lymphocytes of the immune system are the only cells in the body to make antibodies, while developing red blood cells are the only cells that make the oxygen transport protein hemoglobin. The differences between a neuron, a lymphocyte, a liver cell, and a red blood cell depend upon the precise control of gene expression. In each case, the cell is using only some of the genes in its total repertoire. In this chapter, we shall discuss the main ways in which gene expression is controlled in bacterial and eukaryotic cells. Although some mechanisms of control apply to both sorts of cells, eukaryotic cells, through their more complex chromosomal structure, have ways of controlling gene expression that are not available to bacteria. An overview of gene expression. How does an individual cell specify which of its many thousands of genes to express? This decision is an especially important problem for multicellular organisms, because as the animal develops, cell types such as muscle, nerve, and blood cells become different from one another, eventually leading to the wide variety of cell types seen in the adult. Such differentiation arises because cells make and accumulate different sets of RNA and protein molecules. That is, they express different genes. The different cell types of a multicellular organism contain the same DNA. As discussed above, cells have the ability to change which genes they express without altering the nucleotide sequence of their DNA. But how do we know this? If DNA were altered irreversibly during development, the chromosomes of a differentiated cell would be incapable of guiding the development of the whole organism. To test this idea, a nucleus from a skin cell of an adult frog was injected into a frog egg whose own nucleus had been removed. In at least some cases, the egg developed normally into a tadpole, indicating that the transplanted skin cell nucleus cannot have lost any critical DNA sequences. Figure 8.2. Such nuclear transplantation experiments have also been carried out successfully using differentiated cells taken from adult mammals, including sheep, cows, pigs, goats, and mice. And in plants, individual cells removed from a carrot, for example, can be shown to regenerate an entire adult carrot plant. These experiments all show that the DNA in specialized cell types still contains the entire set of instructions needed to form a whole organism. The cells of an organism, therefore, differ not because they contain different genes, but because they express them differently. Different cell types produce different sets of proteins. The extent of the differences in gene expression between different cell types may be roughly gauged by comparing the protein composition of cells in liver, heart, brain, and so on using the technique of two-dimensional gel electrophoresis. See panel 4.6, page 167. Experiments of this kind reveal that many proteins are common to all the cells of a multicellular organism. These housekeeping proteins include the structural proteins of chromosomes, RNA polymerases, DNA repair enzymes, ribosomal proteins, enzymes involving glycolysis and other basic metabolic processes, and many of the proteins that form the cytoskeleton. Each different cell type also produces specialized proteins that are responsible for the cell's distinctive properties. In mammals, for example, hemoglobin is made in reticulocytes, the cells that develop into red blood cells, but it cannot be detected in any other cell type. Many proteins in a cell are produced in such small numbers that they cannot be detected by the technique of gel electrophoresis. A more sensitive technique, called mass spectrometry, figure 445, can be used to detect even rare proteins, and can also provide information about whether the proteins are covalently modified, for example by phosphorylation. Gene expression can also be studied by monitoring the mRNAs that encode proteins, rather than the proteins themselves. Estimates of the number of different mRNA sequences in human cells suggest that at any one time, a typical differentiated human cell expresses perhaps 5,000 to 15,000 genes from a repertoire of about 25,000. It is the expression of a different collection of genes in each cell type that causes the large variations seen in the size, shape, behavior, and function of differentiated cells. A cell can change the expression of its genes in response to external signals. Most of the specialized cells in a multicellular organism are capable of altering their patterns of gene expression in response to extracellular cues. For example, if a liver cell is exposed to a glucocorticoid hormone, a type of steroid, the production of several specific proteins is dramatically increased. Released in the body during periods of starvation or intense exercise, glucocorticoids signal the liver to increase the production of glucose from amino acids and other small molecules. The set of proteins whose production is induced by glucocorticoids includes enzymes such as tyrosine aminotransferase, which helps to convert tyrosine to glucose. When the hormone is no longer present, the production of these proteins drops to its normal level. Other cell types respond to glucocorticoids differently. In fat cells, for example, the production of tyrosine aminotransferase is reduced, while some other cell types do not respond to glucocorticoids at all. These examples illustrate a general feature of cell specialization. Different cell types often respond in different ways to the same extracellular signal. Underlying such adjustments are features of the gene expression pattern that do not change and give each cell type its permanently distinctive character. Gene expression can be regulated at many of the steps in the pathway from DNA to RNA to protein. Differences among the various cell types of an organism depend on the particular genes that the cell express. At what level is the control of gene expression exercised? As we saw in the last chapter, there are many steps in the pathway leading from DNA to protein, and all of them can in principle be regulated. Thus, a cell can control the proteins it makes by 1. Controlling when and how often a given gene is transcribed. 2. Controlling how an RNA transcript is spliced or otherwise processed. 3. Selecting which mRNAs are exported from the nucleus to the cytosol. 4. Selectively degrading certain mRNA molecules. 5. Selecting which mRNAs are translated by ribosomes. Or 6. selectively activating or inactivating proteins after they have been made. Figure 8.3. Gene expression can be regulated at each of these steps, and throughout this chapter we will describe some of the key control points along their pathway from DNA to protein. For most genes, however, the control of transcription, step number 1 in Figure 8.3, is paramount. This makes sense because only transcriptional control can ensure that no unnecessary intermediates are synthesized. So it is the regulation of transcription and the DNA and protein components that determine which genes a cell transcribes into RNA that we address first. How transcriptional switches work. Only 50 years ago, the idea that genes could be switched on and off was revolutionary. This concept was a major advance, and it came originally from the study of how E. coli bacteria adapt to changes in the composition of their growth medium. Many of the same principles apply to eukaryotic cells. However, the enormous complexity of gene regulation in higher organisms, combined with the packaging of their DNA into chromatin, creates special challenges and some novel opportunities for control, as we shall see. We begin with a discussion of the transcription regulators, proteins that control gene expression at the level of transcription. Transcription is controlled by proteins binding to regulatory DNA sequences. Control of transcription is usually exerted at the step at which the process is initiated. In Chapter 7, we saw that the promoter region of a gene attracts the enzyme RNA polymerase and correctly orients the enzyme to begin its task of making an RNA copy of the gene. The promoters of both bacterial and eukaryotic genes include an initiation site, where transcription actually begins, and a sequence of approximately 50 nucleotides that extends upstream from the initiation site, if one likens the direction of transcription to the flow of a river. This region contains sites that are required for the RNA polymerase to bind to the promoter. In addition to the promoter, nearly all genes, whether bacterial or eukaryotic, have regulatory DNA sequences that are used to switch the gene on or off. Some regulatory DNA sequences are as short as 10 nucleotide pairs and act as simple gene switches that respond to a single signal. Such simple switches predominate in bacteria. Other regulatory DNA sequences, especially those in eukaryotes, are very long, sometimes more than 10,000 nucleotide pairs, and act as molecular microprocessors, integrating information from a variety of signals into a command that dictates how often transcription is initiated. Regulatory DNA sequences do not work by themselves. To have any effect, these sequences must be recognized by proteins called transcription regulators, which bind to the DNA. It is a combination of a DNA sequence and its associated protein molecules that acts as a switch to control transcription. The simplest bacterium codes for several hundred transcription regulators, each of which recognizes a different DNA sequence and thereby regulates a distinct set of genes. Humans make many more, several thousand, signifying the importance and complexity of this form of gene regulation in producing a complex organism. Proteins that recognize a specific DNA sequence do so because the surface of the protein fits tightly against the special surface features of the double helix in that region. These features will vary depending on the nucleotide sequence, and thus different proteins will recognize different nucleotide sequences. In most cases, the protein inserts into the major groove of the DNA helix, see Figure 5.7, and makes a series of molecular contacts with the base pairs. The protein forms hydrogen bonds, ionic bonds, and hydrophobic interactions with the edges of the bases, usually without disrupting the hydrogen bonds that hold the base pairs together. Figure 8.4. Although each individual contact is weak, the 20 or so contacts that are typically formed at the protein-DNA interface combine to ensure that the interaction is both highly specific and very strong. Indeed, Protein-DNA interactions are among the tightest and most specific molecular interactions known in biology. Although each example of protein-DNA recognition is unique in detail, many of the proteins responsible for gene regulation recognize DNA through one of several structural motifs. These fit into the major groove of the DNA double helix and form tight associations with a short stretch of DNA base pairs. The DNA binding motifs, shown in Figure 8.5, the homeodomain, the zinc finger, and the leucine zipper, are found in transcription regulators that control the expression of thousands of different genes in virtually all eukaryotic organisms. Frequently, DNA binding proteins bind in pairs, dimers, to the DNA helix. Dimerization roughly doubles the area of contact with the DNA, thereby greatly increasing the strength and specificity of the protein-DNA interaction. Because two different proteins can pair in different combinations, dimerization also makes it possible for many different DNA sequences to be recognized by a limited number of proteins. Transcription switches allow cells to respond to changes in the environment. The simplest and most completely understood examples of gene regulation occur in bacteria and in the viruses that infect them. The genome of the bacterium E. coli consists of a single circular DNA molecule of about 4.6 times 10 to the power of 6 nucleotide pairs. This DNA encodes approximately 4,300 proteins, although only a fraction of these are made at any one time. Bacteria regulate the expression of many of their genes according to the food sources that are available in the environment. For example, in E. coli, 5 genes code for enzymes that manufacture the amino acid tryptophan. These genes are arranged in a cluster on the chromosome and are transcribed from a single promoter as one long mRNA molecule from which the 5 proteins are translated. Figure 8.6. When tryptophan is presented in the surroundings and enters the bacterial cell, these enzymes are no longer needed and their production is shut off. This situation arises, for example, when the bacterium is in the gut of a mammal and has just eaten a meal rich in protein. These 5 coordinately expressed genes are part of an operon, a set of genes that are transcribed into a single mRNA. Operons are common in bacteria, but are not found in eukaryotes, where genes are transcribed and regulated individually. See Figure 736. We now understand in considerable detail how the tryptophan operon functions. Within the promoter is a short DNA sequence, 50 nucleotides in length, that is recognized by a transcription regulator. When this protein binds to this nucleotide sequence, termed the operator, it blocks access of RNA polymerase to the promoter. This prevents transcription of the operon and production of the tryptophan-producing enzymes. The transcription regulator is known as a tryptophan repressor, and it is controlled in an ingenious way. The repressor can bind to DNA only if it has also bound several molecules of the amino acid tryptophan. Figure 8.7. The tryptophan repressor is an allosteric protein. See Figure 437. The binding of tryptophan causes a subtle change in its three-dimensional structure so that the protein can bind to the operator sequence. When the concentration of free tryptophan in the cell drops, the repressor no longer binds tryptophan and thus no longer binds to DNA, and the tryptophan operon is transcribed. The repressor is thus a simple device that switches production of a set of biosynthetic enzymes on and off according to the availability of the end product of the pathway that the enzymes catalyze. The bacterium can respond very rapidly to the rise in tryptophan concentration because the tryptophan repressor protein itself is always present in the cell. The gene that encodes it is continuously transcribed at a low level, so that a small amount of the repressor protein is always being made. Such unregulated gene expression is known as constitutive gene expression. Repressors turn genes off, activators turn them on. The tryptophan repressor, as its name suggests, is a repressor protein. In its active form, it switches genes off, or represses them. Some bacterial transcription regulators do the opposite. They switch genes on, or activate them. These activator proteins work on promoters that, in contrast to the promoter for the tryptophan operator, are, on their own, only marginally able to bind and position RNA polymerase. They may, for example, be recognized only poorly by the polymerase. However, these poorly functioning promoters can be made fully functional by activator proteins that bind to a nearby site on the DNA and contact the RNA polymerase to help it initiate transcription. Figure 8.8. In some cases, a bacterial transcription regulator can repress transcription at one promoter and activate transcription at another. Whether the regulatory protein acts as an activator or repressor depends, in large part, on exactly where the regulatory sequences to which it binds are located with respect to the promoter. Like the tryptophan repressor, activator proteins often have to interact with a second molecule to be able to bind DNA. For example, the bacterial activator protein CAP has to bind cyclic AMP CAMP before it can bind to DNA. Genes activated by CAP are switched on in response to an increase in intracellular CAMP concentration, which signals to the bacterium that glucose, its preferred carbon source, is no longer available. As a result, CAP drives the production of enzymes capable of degrading other sugars. An activator and a repressor control the Lac operon. In many instances, the activity of a single promoter is controlled by two different transcription regulators. The Lac operon in E. coli, for example, is controlled by both the Lac repressor and the activator protein CAP. The Lac operon encodes proteins required to import and digest the disaccharide lactose. In the absence of glucose, CAP switches on genes that allow the cell to utilize alternative sources of carbon, including lactose. It would be wasteful, however, for CAP to induce expression of the Lac operon when lactose is not present. Thus, the Lac repressor shuts off the operon in the absence of lactose. This arrangement enables the control region of the Lac operon to integrate two different signals, so that the operon is highly expressed only when two conditions are met. Lactose must be present and glucose must be absent. Figure 8.9. This genetic circuit thus behaves like a switch that carries out a logic operation in a computer. When lactose is present and glucose is absent, the cell executes the appropriate program. In this case, transcription of the genes that permit the uptake and utilization of lactose. The elegant logic of the Lac operon first attracted the attention of biologists more than 50 years ago. The molecular basis of the switch was uncovered by a combination of genetics and biochemistry, providing the first insight into how gene expression is controlled. In a eukaryotic cell, similar gene regulatory devices are combined to generate increasingly complex circuits. Indeed, the developmental program that takes a fertilized egg to adulthood can be viewed as an exceedingly complex circuit composed of simple components, like those that control the Lac and tryptophan operons. Eukaryotic transcription regulators control gene expression from a distance. Eukaryotes, too, use transcription regulators, both activators and repressors, to regulate the expression of their genes. The DNA sites to which eukaryotic gene activators bound were originally termed enhancers, because their presence dramatically enhanced or increased the rate of transcription. It was surprising to biologists when in 1979 it was discovered that these activator proteins could enhance transcription even when they are bound thousands of nucleotide pairs away from a gene's promoter. They also work when bound either upstream or downstream from the gene. These observations raised several questions. How do enhancer sequences and the proteins bound to them function over such long distances? How do they communicate with a promoter? Many models for this action at a distance have been proposed, but the simplest of these seems to apply in most cases. The DNA between the enhancer and the promoter loops out to allow eukaryotic activator proteins to directly influence events that take place at the promoter. Figure 8.10. The DNA thus acts as a tether, causing a protein bound to an enhancer even thousands of nucleotide pairs away to interact with the proteins in the vicinity of the promoter, including RNA polymerase II and the general transcription factors. See Figure 7.12. Often, additional proteins serve to link the distantly bound transcription regulators to these proteins at the promoter. The most important is a large complex of proteins known as mediator. See Figure 8.10. One of the ways in which eukaryotic activator proteins function is by aiding in the assembly of the general transcription factors and RNA polymerase at the promoter. Eukaryotic repressor proteins do the opposite. They decrease transcription by preventing or sabotaging the assembly of the same protein complex. In addition to promoting or repressing the assembly of a transcription initiation complex, eukaryotic transcription regulators have an additional mechanism of action. They attract proteins that modulate chromatin structure and thereby affect the accessibility of the promoter to the general transcription factors and RNA polymerase, as we discuss next. Packing of promoter DNA into nucleosomes affects initiation of transcription. Initiation of transcription in eukaryotic cells must also take into account the packaging of DNA into chromosomes. As we saw in Chapter 5, the genetic material in eukaryotic cells is packed into nucleosomes, which in turn are folded into higher-order structures. How do transcription regulators, general transcription factors, and RNA polymerase gain access to such DNA? Nucleosomes can inhibit the initiation of transcription if they are positioned over a promoter, probably because they physically block the assembly of the general transcription factors or RNA polymerase on the promoter. In fact, such chromatin packaging may have evolved in part to prevent leaky gene expression, initiation of transcription in the absence of the proper activator proteins. In eukaryotic cells, activator and repressor proteins exploit chromatin structure to help turn genes on and off. As we saw in Chapter 5, chromatin structure can be altered by chromatin remodeling complexes and by enzymes that covalently modify the histone proteins that form the core of the nucleosome. See Figure 5.27 and 5.28. Many gene activators take advantage of these mechanisms by recruiting these proteins to promoters. Figure 8.11. For example, many transcription activators attract histone acetylases, which attach an acetyl group to selected lysines in the tail of histone proteins. This modification alters chromatin structure, probably allowing greater accessibility to the underlying DNA. Moreover, the acetyl groups themselves are recognized by proteins that promote transcription, including some of the general transcription factors. Likewise, gene repressor proteins can modify chromatin in ways that reduce the efficiency of transcription initiation. For example, many repressors attract histone deacetylases, enzymes that remove the acetyl groups from histone tails, thereby reversing the positive effects that acetylation has on transcription initiation. Although some eukaryotic repressor proteins work on a gene-by-gene basis, others can orchestrate the formation of large swaths of transcriptionally inactive chromatin containing many genes. As discussed in Chapter 5, these transcription-resistant regions of DNA include the heterochromatin found in interphase chromosomes and the entire X chromosome in female mammals. The molecular mechanisms that create specialized cell types. All cells must be able to switch genes on and off in response to signals in their environments, but the cells of multicellular organisms have evolved this capacity to an extreme degree and in highly specialized ways to form an organized array of differentiated cell types. In particular, once a cell in a multicellular organism becomes committed to differentiate into a specific cell type, the choice of fate is generally maintained through many subsequent cell generations. This means that the changes in gene expression, which are often triggered by a transient signal, must be remembered. The phenomenon of cell memory is a prerequisite for the creation of organized tissues and for the maintenance of stably differentiated cell types. In contrast, the simplest changes in gene expression in both eukaryotes and bacteria are often only transient. The tryptophan repressor, for example, switches off the tryptophan genes in bacteria only in the presence of tryptophan. As soon as the amino acid is removed from the medium, the genes are switched back on, and the descendants of the cell will have no memory that their ancestors had been exposed to tryptophan. In this section, we discuss some of the special features of transcriptional regulation that are found in multicellular organisms. Our focus will be on how these mechanisms create and maintain the specialized cell types that give a worm, a fly, or a human its distinctive characteristics. Eukaryotic genes are regulated by combinations of proteins. Because eukaryotic transcription regulators can control transcription initiation when bound to DNA, many base pairs away from the promoter, the nucleotide sequences that control the expression of a gene can be spread over long stretches of DNA. In animals and plants, it is not unusual to find regulatory DNA sequences of a gene dotted over tens of thousands of nucleotide pairs, although much of this DNA serves as spacer sequence and is not directly recognized by the transcription regulators. So far in this chapter, we have treated transcription regulators as though each functions individually to turn a gene on or off. While this idea holds true for many simple bacterial activators and repressors, most eukaryotic transcription regulators work as part of a committee of regulatory proteins, all of which are necessary to express a gene in the right cell, in response to the right conditions, at the right time, and in the required amount. The term combinatorial control refers to the way that groups of regulatory proteins work together to determine the expression of a single gene. We saw a simple example of such regulation by multiple signals when we discussed the Bacterial Lac Operon. See Figure 8.9. In eukaryotes, the regulatory inputs have been amplified, and a typical gene is controlled by dozens of transcription regulators. Figure 8.12. Often, some of these regulatory proteins are repressors and some are activators. The molecular mechanisms by which the effects of all of these proteins are added up to determine the final level of expression for a gene are only now beginning to be understood. An example of such a complex regulatory system, one that participates in the development of a fruit fly from a fertilized egg, is described in How We Know, pages 282 to 284. The expression of different genes can be coordinated by a single protein. In addition to being able to switch individual genes on and off, all organisms, whether prokaryote or eukaryote, need to coordinate the expression of different genes. When a eukaryotic cell receives a signal to divide, for example, a number of hitherto unexpressed genes are turned on together to set in motion the events that lead eventually to cell division, discussed in Chapter 18. One way in which bacteria coordinate the expression of a set of genes is by having them clustered together in an operon under the control of a single promoter. See Figure 8.6. This is not the case in eukaryotes, in which each gene is transcribed and regulated individually. So how do eukaryotes coordinate gene expression? In particular, given that a eukaryotic cell uses a committee of transcription regulators to control each of its genes, how can it rapidly and decisively switch whole groups of genes on or off? The answer is that even though control of gene expression is combinatorial, the effect of a single transcription regulator can still be decisive in switching any particular gene on or off. simply by completing the combination needed to activate or repress that gene. This is like dialing in the final number of a combination lock. The lock will spring open if the other numbers have been previously entered. Just as the same number can complete the combination for different locks, the same protein can complete the combination for several different genes. As long as different genes contain DNA sequences recognized by the same transcription regulator, they can be switched on or off together as a unit. An example of this style of regulation in humans is seen with the glucocorticoid receptor protein. In order to bind to regulatory sites in DNA, this transcription regulator must first form a complex with a molecule of a glucocorticoid hormone, for example, cortisol. See Table 16.1, page 535. In response to glucocorticoids, liver cells increase the expression of many different genes, one of which encodes the enzyme tyrosine aminotransferase, as discussed earlier. These genes are all regulated by the binding of the glucocorticoid hormone receptor complex to a regulatory sequence in the DNA of each gene. When the body has recovered and the hormone is no longer present, the expression of all of these genes drops to its normal level. In this way, a single transcription regulator can control the expression of many different genes. Figure 8.17. Combinatorial control can create different cell types. The ability to switch many different genes on or off using just one protein is not only useful in the day-to-day regulation of cell function, it is also one of the means by which eukaryotic cells differentiate into particular types of cells during embryonic development. A striking example of the effect of a single transcription regulator on differentiation comes from studying the development of muscle cells. A mammalian skeletal muscle cell is a highly distinctive cell type. It is typically an extremely large cell that is formed by the fusion of many muscle precursor cells called myoblasts. The mature muscle cell is distinguished from other cells by the production of a large number of characteristic proteins, such as the actin and myosin that make up the contractile apparatus, discussed in Chapter 17, as well as the receptor proteins and ion channel proteins in the cell membranes that make the muscle cells sensitive to nerve stimulation. Genes encoding these muscle-specific proteins are all switched on coordinately at the myoblasts as the myoblasts begin to fuse. Studies of muscle cells differentiating in culture have identified key transcription regulators, expressed only in potential muscle cells, that coordinate gene expression and thus are crucial for muscle cell differentiation. These regulators activate the transcription of the genes that code for the muscle-specific proteins by binding to specific DNA sequences present in their regulatory regions. These key transcription regulators can convert non-muscle cells to myoblasts by activating the changes in gene expression typical of differentiating muscle cells. For example, when one of these regulators, MyoD, is artificially expressed in fibroblasts cultured from skin connective tissue, the fibroblasts start to behave like myoblasts and fuse to form muscle-like cells. The dramatic effect of expressing the MyoD gene in fibroblasts is shown in Figure 8.18. It appears that the fibroblasts, which are derived from the same broad class of embryonic cells as muscle cells, have already accumulated many of the other necessary transcription regulators required for the combinatorial control of the muscle-specific genes, and that addition of MyoD completes the unique combination that directs the cells to become muscle. Some other cell types fail to be converted to muscle by the addition of MyoD. These cells presumably have not accumulated the other required transcription regulators during their developmental history. How the accumulation of different transcription regulators can lead to the generation of different cell types is illustrated schematically in Figure 8.19. This figure also illustrates how, thanks to the possibilities of combinatorial control and shared regulatory DNA sequences, a limited set of transcription regulators can control the expression of a much larger number of genes. The conversion of one cell type, fibroblast, to another, muscle, by a single transcription regulator emphasizes one of the most important principles discussed in this chapter. The dramatic differences between cell types, such as size, shape, and function, are produced by differences in gene expression. Stable patterns of gene expression can be transmitted to daughter cells. As discussed earlier in this chapter, once a cell in a multicellular organism has become differentiated into a particular cell type, it will generally remain differentiated, and all its progeny cells will remain that same cell type. Some highly specialized cells never divide again once they have differentiated, for example, skeletal muscle cells and neurons, but many other differentiated cells, such as fibroblasts, smooth muscle cells, and liver cells, hepatocytes, will divide many times in the life of an individual. All of these cell types give rise only to cells like themselves when they divide. Smooth muscle does not give rise to liver cells, nor liver cells to fibroblasts. This preservation of cellular identity means that the changes in gene expression that give rise to a differentiated cell must be remembered and passed on to its daughter cells through all subsequent cell divisions. For example, in the cells illustrated in Figure 819, the production of each transcription regulator, once begun, has to be perpetuated in the daughter cells of each cell division. How might this be accomplished? Cells have several ways of ensuring that their daughters remember what kind of cells they are supposed to be. One of the simplest is through a positive feedback loop, where a key transcription regulator activates transcription of its own gene in addition to that of other cell type specific genes. Figure 820. The MyoD protein discussed earlier functions in such a positive feedback loop. Another way of maintaining cell type is through the faithful propagation of a condensed chromatin structure from parent to daughter cell. We saw an example of this in Figure 530, where the same X chromosome is inactive through many cell generations. A third way in which cells can transmit information about gene expression to their progeny is through DNA methylation. In vertebrate cells, DNA methylation occurs exclusively on cytosine bases. Figure 821. This covalent modification of cytosines generally turns off genes by attracting proteins that block gene expression. DNA methylation patterns are passed on to progeny cells by the action of an enzyme that copies the methylation pattern on the parent DNA strand to the daughter DNA strand immediately after replication. Figure 822. Because each of these mechanisms, positive feedback loops, certain forms of condensed chromatin, and DNA methylation, transmits information from parent to daughter cell without altering the actual nucleotide sequence of the DNA, they are considered forms of epigenetic inheritance. See page 192. The formation of an entire organ can be triggered by a single transcription regulator. We have seen that even though combinatorial control is the norm for eukaryotic genes, a single transcription regulator can be decisive in switching a whole set of genes on or off, and can convert one cell type into another. A dramatic extension of this principle comes from studies of eye development in Drosophila, mice, and humans. Here, a transcription regulator called A in flies and PAX6 in vertebrates is crucial for eye development. When expressed in the proper type of cell, A can trigger the formation of not just a single cell type, but a whole organ, the eye, composed of different types of cells, all properly organized in three-dimensional space. The best evidence for the action of A comes from experiments in fruit flies in which the A gene is artificially expressed early in development in cells that normally go on to form legs. This abnormal gene expression causes eyes to develop in the middle of the legs. Figure 823. The Drosophila eye is composed of thousands of cells, and how the eye protein coordinates the specification of each cell in the eye is an actively studied topic in developmental biology. Here, we shall simply note that eye directly controls the expression of many genes by binding to DNA sequences in the regulatory regions. Some of the genes controlled by eye encode additional transcription regulators that in turn control the expression of other genes. Moreover, some of these regulators act back on eye itself to create a protein feedback loop that ensures the continued production of the eye protein. So the action of just one transcription regulator can produce a cascade of regulators whose combined actions lead to the formation of an organized group of many different types of cells. One can begin to imagine how, by repeated applications of this principle, a complex organism is built piece by piece. Post-transcriptional controls. We have seen that transcription regulators control gene expression by switching on or off transcription initiation. The vast majority of genes in all organisms are regulated in this way, but additional points of control can come into play later in the pathway from DNA to protein, giving cells a further opportunity to manage the amount of gene product that is made. These post-transcriptional controls, which operate after RNA polymerase has bound to a gene's promoter and started to synthesize RNA, are crucial for the regulation of many genes. In Chapter 7, we described one type of post-transcriptional control, alternative splicing, which allows different forms of a protein to be made in different tissues. Figure 721. Here we discuss a few more examples of the many ways in which cells can manipulate gene expression after transcription has begun. Ribose switches provide an economical solution to gene regulation. The mechanisms for controlling gene expression we have described thus far all involve the participation of a regulatory protein, but scientists have recently discovered a number of mRNAs that can regulate their own transcription and translation. These self-regulating mRNAs contain ribose switches, short sequences of RNA that change their conformation when bound to small molecules such as metabolites. Many ribose switches have been discovered, and each recognizes a specific small molecule. The conformational change that is driven by the binding of that molecule can regulate gene expression. Figure 824. This mode of gene regulation is particularly common in bacteria, where ribose switches sense key small metabolites in the cell and adjust gene expression accordingly. Ribose switches are perhaps the most economical examples of gene control devices, because they bypass the need for regulatory proteins altogether. The fact that short sequences of RNA can form such highly efficient gene control devices offers further evidence that, before modern cells arose, a world run by RNAs may have reached a high level of sophistication. See pages 261 to 264. The untranslated regions of mRNAs can control their translation. Once an mRNA has been synthesized, one of the most common ways of regulating how much of its protein product is made is to control the initiation of translation. Although the details of translation initiation differ between eukaryotes and bacteria, both use the same basic strategies for regulating gene expression at this step. Bacterial mRNAs contain a short ribosome binding sequence located a few nucleotides upstream of the AUG codon where translation begins. The recognition sequence forms base pairs with the RNA in the small ribosomal subunit, correctly positioning the initiating AUG codon within the ribosome. Because this interaction is needed for efficient translation initiation, it provides an ideal target for translational control. By blocking or exposing the ribosome recognition sequence, the bacterium can either inhibit or promote the translation of an mRNA. Figure 825. Eukaryotic mRNAs possess a 5' cap that helps guide the ribosome to the first AUG, the codon where translation will start. See Figure 735. In eukaryotic cells, repressors can inhibit translation initiated by binding to specific RNA sequences in the 5' untranslated region of the mRNA and keeping the ribosome from finding the first AUG. When conditions change, the cell can inactivate the repressor and thereby increase translation of the mRNA. Small regulatory RNAs control the expression of thousands of animal and plant genes. As we saw in Chapter 7, RNAs perform many critical tasks in cells. In addition to acting as intermediate carriers of genetic information, they play key structural and catalytic roles, particularly in protein synthesis. See pages 253 to 254. But a recent series of striking discoveries has revealed that non-coding RNAs, those that do not direct the production of a protein product, are far more prevalent than previously imagined and play unanticipated widespread roles in regulating gene expression. One particularly important type of non-coding RNA found in plants and animals is called microRNA, mRNA. Humans, for example, produce more than 400 different mRNAs, which seem to regulate at least 1/3 of all protein-coding genes. These short regulatory RNAs control gene expression by base pairing with specific mRNAs and controlling their stability and their translation. Like other non-coding RNAs, such as tRNA and rRNA, the precursor mRNA transcript undergoes a special type of processing to yield the mature mRNA. This mRNA is then assembled with specialized proteins to form an RNA-induced silencing complex, RISC. The RISC patrols the cytoplasm, searching for mRNAs that are complementary to the mRNA it carries. Figure 826. Once a target mRNA forms base pairs with a mRNA, it is destroyed immediately by a nuclease present within the RISC, or else its translation is blocked and it is delivered to a region of the cytoplasm where other nucleases will eventually degrade it. Once the RISC has taken care of an mRNA molecule, it is released and is free to seek out additional mRNA molecules. Thus, a single mRNA, as part of a RISC, can eliminate one mRNA molecule after another, thereby efficiently blocking production of the protein that the mRNA encodes. Two features of mRNA make them especially useful regulators of gene expression. First, a single mRNA can regulate a whole set of different mRNAs so long as the mRNAs carry a common sequence. These sequences are often located in their 5' and 3' untranslated regions. In humans, some individual mRNAs control hundreds of different mRNAs in this manner. Second, a gene that encodes a mRNA occupies relatively little space in the genome compared with one that encodes a transcription regulator. Indeed, their small size is one reason that mRNAs are discovered only recently. Although we are only beginning to understand the full impact of mRNAs, it is clear that they represent a critical part of the cell's equipment for regulating the expression of its genes. Rna interference destroys double-stranded foreign RNAs. Some of the proteins that process and package mRNAs also serve as a cell defense mechanism. They orchestrate the destruction of foreign RNA molecules, specifically those that are double-stranded. Many viruses and transposable genetic elements produce double-stranded RNA sometime in their life cycles. This targeted RNA degradation mechanism, called Rna Interference, RNAi, helps to keep these potentially dangerous invaders in check. The presence of foreign double-stranded RNA in the cell triggers RNAi by first attracting a protein complex containing a nuclease called dicer. dicer cleaves the double-stranded RNA into short fragments, approximately 23 nucleotide pairs in length, called small interfering RNAs, cRNAs. These short, double-stranded RNAs are then incorporated into RISCs, the same complexes that can carry mRNAs. The RISC discards one strand of the cRNA duplex and uses the remaining single-stranded RNA to locate a complementary foreign RNA molecule, figure A27. This target RNA molecule is then rapidly degraded, leaving the RISC free to search out more of the same foreign RNA molecule. RNAi is found in a wide variety of organisms, including single-celled fungi, plants, and worms, indicating that it is evolutionarily ancient. In some organisms, including plants, the RNAi activity can spread from tissue to tissue by the movement of RNA between cells. This RNA transfer allows the entire plant to become resistant to a virus after only a few of its cells have been infected. In a broad sense, the RNAi response resembles certain aspects of the human immune system. In both cases, an infectious organism elicits the production of attack molecules, either cRNAs or antibodies, that are custom designed to inactivate the invader and thereby protect the host. Scientists can use RNA interference to turn off genes. The discovery of mRNAs, cRNAs, and the mechanism of RNAi has been greeted with great enthusiasm. In a practical sense, RNAi has become a powerful experimental tool that allows scientists to inactivate almost any gene in cultured cells, or in some cases, a whole plant or animal. We discuss how this method is being used to help determine the function of individual genes in Chapter 10. At the same time, RNAi shows real potential as a powerful new approach for treating human disease. Because many human disorders result from the inappropriate expression of genes, the ability to turn these genes off by introducing complementary cRNA molecules holds great medical promise. Finally, the discovery that RNAs play such a key role in controlling gene expression expands our understanding of the types of regulatory networks that cells have at their command. One of the great challenges of biology in this century will be to determine how these networks cooperate to specify the development of complex organisms, including ourselves. Essential concepts. A typical eukaryotic cell expresses only a fraction of its genes, and the distinct types of cells in multicellular organisms arise because different sets of genes are expressed as cells differentiate. Although all of the steps involved in expressing a gene can in principle be regulated, for most genes, the initiation of transcription is the most important point of control. The transcription of individual genes is switched on and off in cells by transcription regulators. These act by binding to short stretches of DNA called regulatory DNA sequences. Although each transcription regulator has unique features, most bind to DNA using one of a small number of structural motifs. The precise amino acid sequence that is folded into the DNA binding motif determines the particular DNA sequence that is recognized. In bacteria, transcription regulators usually bind to regulatory DNA sequences close to where RNA polymerase binds. They can either activate or repress transcription of the gene. In eukaryotes, regulatory DNA sequences are often separated from the promoter by many thousands of nucleotide pairs. Eukaryotic transcription regulators act in two fundamental ways. One, they can directly affect the assembly process of RNA polymerase and the general transcription factors at the promoter. And two, they can locally modify the chromatin structure of promoter regions. In eukaryotes, the expression of a gene is generally controlled by a combination of transcription regulators. In multicellular plants and animals, the production of different transcription regulators in different cell types ensures the expression of only those genes appropriate to the particular type of cell. Cells in multicellular organisms have mechanisms that enable their progeny to remember what type of cell they should be. A single transcription regulator, if expressed in the appropriate precursor cell, can trigger the formation of a specialized cell type or even an entire organ. Cells can also regulate gene expression by controlling events that occur after transcription has begun. Many of these mechanisms rely on RNA molecules that can influence their own transcription or translation. MicroRNAs, mRNAs, control gene expression by base pairing with specific mRNAs and regulating their stability and their translation. Cells have a defense mechanism for destroying foreign double-stranded RNAs, many of which are produced by viruses. Scientists can take advantage of this mechanism, called Rna interference, to inactivate genes of interest by simply injecting cells with double-stranded RNAs that target the mRNAs produced by those genes.